Huntington's disease was discovered in 1872, but it wasn't until 1993 that it was recognized as a mutation on the fourth chromosome. It is characterized by CAG repeats coding for glutamic acid. The normal range for these repeats is 10 to 34. Any number greater than 39 results in Huntington's disease. Huntington's disease has its origins in Western Europe. We are seeing Caucasians being the most affected population, whereas Asian populations are least affected. The prevalence of Huntington's disease has been increasing by 15 to 20% per decade. This disease is a neurodegenerative disease characterized by mood fluctuations, memory impairments, involuntary muscular movements, and eventually death. Huntington's disease has an average age of onset of about 30 to 45 years old. Being an autosomal dominant disease, there's a 50% chance of passing it on to your offspring if you're affected. The study Sex-Specific Effects of the Huntington Gene on Normal Neurodevelopment by Lee relates Huntington's disease to human intelligence. It shows that CAG repeats seen in the Huntington gene below 39 are associated with higher intelligence and higher IQ. Increases in the number of repeats in the normal range, which is 10 to 34, are beneficial in terms of structure and function of the human brain. The study Evolution and Function of CAG polyglutamine repeats in protein-protein interaction networks by Schaefer goes over the range of CAG repeats related to Huntington's disease and that gene. The wild-type range is between 2 and 26 repeats. The range where the genotype is unstable and can potentially result in Huntington's disease is between 29 and 35 repeats. Late onset of Huntington's disease is related to 36 to 39 repeats, and a guarantee of developing Huntington's disease and eventually dying is above 40 repeats. The article Biologists Link Huntington's Disease to Health Benefits in Young People by Tufts University shows the relationship between the Huntington gene and an increase in p53 protein. p53 protein is important because it reduces cancer growths and increases the immune system function. The study Huntington's Disease, A Clinical Review by Raymond emphasizes the importance of the unstable range of CAG repeats from 29 to 35. This range is important because it's prone to elongation during transcription. This means that environmental factors could potentially affect the transcription of this gene and lead to the development of Huntington's disease. The Epigenetic Mechanisms and Genome Stability by Putrieri discusses the CAG expansion seen in Huntington's disease. This expansion is characterized by the anticipation phenomenon, which means repeat expansion increases every generation. This can be related to the other studies, and that environmental stressors can cause unstable CAG repeats to increase to a detrimental level, causing disease. Traditional medicine approaches to treating Huntington's disease focus on screening for prevention, since there aren't any known effective treatments as of today. Early detection of Huntington's disease allows for counseling and therapy before the onset of symptoms and helps to manage the cognitive and physical disabilities that come with the development of disease. This approach focuses on mechanism of gene transcription and the number of repeats that will cause the disease in an individual. The evolutionary medicine approach to Huntington's disease takes a much broader perspective, combining biology, medicine, and human evolution. From an evolutionary standpoint, Huntington's disease has potential benefits, can be seen as a trade-off in human evolution, is related to reproductive fitness, and has roots in epigenetics. The Huntington gene has beneficial origins because CAG repeats below 39, which is the disease threshold, are associated with increased intelligence and brain complexity. An increase in intelligence results in increased learning and communication systems. Intelligence is the basis for human survival and reproduction and allows adaptation to novel environments. This is partially what led to human expansion across the globe and is why we are such a successful species. The Huntington gene is also beneficial in terms of mutation suppression. It is associated with an increase in the p53 protein. The p53 protein is important because it prevents cancers, increases immune system function, and results in better overall health. This would help humans to survive in a harsh environment full of many microbes. Huntington's disease can be seen as a trade-off for intelligence because the increase in CAG repeats is correlated with an increase in intelligence within the normal range of repeats on the Huntington gene. However, in terms of Huntington's disease, these CAG repeats have surpassed that normal range. In general, intelligence and the human ability to learn and communicate are correlated with one another and increase the survival of the human race, so this gene continues to be selected for, despite the fact that it has the potential to cause disease past a certain number of repeats. Disease may occur when the number of repeats surpasses 40, but does not harm reproductive fitness because the detrimental effects of Huntington's disease do not impact an individual until they are past their prime reproductive years. This means Huntington's disease remains in the population and continues to be passed down from one generation to the next. Huntington's disease can be seen as a trade-off for reproduction. This is because the Huntington gene is correlated with an increase in the p53 protein. The p53 protein increases immune system function and decreases the prevalence of cancer in people with the Huntington gene. Because of this increase in health in individuals with the gene, they have a greater level of survival and therefore reproductive success. This brings us to the trade-off of health versus longevity. Due to their greater health from having the Huntington gene, they have a greater ability to produce offspring and contribute their genetic material to future generations. However, they're trading off their longevity for this ability to give future generations genetic material. Huntington's disease can be related to epigenetics in terms of the effect the environment has on gene expression. Stressful environmental conditions can increase mutation rates. Repeat mutations, therefore, can be seen as an adaptive response to a stressful environment to bring about novel traits and help that organism survive. The unstable region of 29 to 34 repeats in the Huntington gene is subject to environmental stressors, which can lead to mutation and potential elongation of the gene, leading to Huntington's disease in generations that may not have had it before. The increase of disease prevalence we see with Huntington's disease today could potentially be due to the chronic stress of modern society and social hierarchies. Future evolutionary medicine approaches to studying Huntington's disease could involve studying the effect stress has on gene expansion, as well as possibly using epigenetics to come up with a treatment for Huntington's disease. It would be interesting to investigate chronic stress and Huntington gene expansion in terms of evaluating historical populations where Huntington's disease is known to be prevalent and seeing how the prevalence of Huntington's disease changes during times of great environmental stress, such as famine. It would also be interesting to use epigenetics as a mechanism of creating a genetic treatment for Huntington's disease. Gene transcription is known to be affected by methylation and acetylation. Methylation tends to reduce transcription of the Huntington gene, and it would be interesting to see if this could be done in a laboratory on people with gene repeats greater than 40. A treatment involving epigenetics and decreased transcription of the Huntington gene could potentially increase the longevity of people who have this disease.